So now, time for the final part of our third chapter, which is transport in plants. Vascular plants, the evolutionary movement onto land, involve the differentiation of plant body into roots and shoots. Vasculature transport nutrients throughout the plant. This transport may occur over long distances. Transport in vascular plants occur in three scales: transport of water and solutes by individual cells, such as root hairs; short distance transport from substances from cell to cell at the levels of tissues and organs; long distance transport within xylem and phloem at the level of the whole plant. It all starts with the roots that absorb water and dissolve mineral from the soil. Water and minerals are transported upward from the roots to shoots as xylem sap. Transpiration, or the loss of water from leaves, creates a force within leaves that pulls xylem sap upward. Through stomata, leaves take in CO2 and expel O2. The CO2 provides carbon for photosynthesis. Some O2 produced by photosynthesis is used in cellular respiration. Sugars are produced by photosynthesis in the leaves. They are transported as phloem sap to roots and other parts of the plant. Roots exchange gases with the air spaces of soil, taking in O2 and discharging CO2. In cellular respiration, O2 supports the breakdown of sugars. The selective permeability of the plasma membrane controls movement of solutes into and out of cell. So all of these would involve specific transport proteins. First of all, you have the proton pumps. So proton pump would create a hydrogen ion gradient. So inside of the cell, you have very little H plus or hydrogen ions. Outside of the cell, you need it to get out. Why is it? So that the outer part will be higher than the inner. So this actually causes the outer part of the cell to be much more positive compared to the inner. Okay, as you can see, more H plus, more positive. So to actually balance it, it's actually due to the transport of many different solutes. This balance must be maintained. Outer part must be positive compared to the inner, so that there will always be a gradient. So this is this cation ion called K plus or potassium must be obtained due to the membrane potential. If there is no H plus, there is a lot outside, there will be no gradient, meaning that K cannot get in. So you need it so that the important ions can get in. Co-transport. In co-transport, a transport protein couples the transport of one solute with the passage to another. So meaning that if NO2 wants to get in, it must also be joined with H plus. So this type of uptake also is responsible for sucrose, which is sugar. So sugar, it cannot get to the cells by itself, so it needs to be escorted by the H plus, which is the hydrogen ions. So through this gradient, sucrose can get in. In 1895, the Irish plant physiologists H.A. Dixon and J. Joly proposed that the water is pulled up by tension from above, which is called the negative pressure. So water is continually being lost from the leaves by transpiration. So both Dixon and Joly believe that loss of water in the leaves exerts a pull in the water in the xylem ducts and draws more water into the leaf. But even the best vacuum pump can only pull water out to a height of only 34 feet or so, because the calm water that high will exert a pressure of 15 pounds per inch square, which is 103 kilopascals. So the 103 kilopascal is counterbalanced by the pressure of the atmosphere. So at that point, they cannot go any further. So how can water be drawn to the top of the sequoia? Sequoia is a very large tree, 330 feet in height. Taking all factors into account, upward of at least 270 pounds per inch is probably needed. So what is the answer of this dilemma? The answer would be lying in the cohesion of water molecules. Cohesion will be the ability of water to cling to each other through the hydrogen bonds that they form. Water potential. To survive, plants must balance water uptake and loss. So you must remember that osmosis is the passive transport of water across the membrane. So water potential will be the measurement that combines the effects of solute concentration and physical pressure to the presence of plant cell wall and determines the direction of movement of water. So as you can remember, water flows from the region of high water potential to low water potential. Plasmolysis. If a plastic cell, which is not firm, is placed in an environment with high solute concentration, it is called hypotonic. So the cell will lose water and become plasmolysed. If the same plastic cell is placed in a solution with lower solute concentration, it is hypotonic. It means that the cell will gain water and become turgid. Healthy plant cells are turgid most of the time. Turgor loss in plants will cause wilting, which can be reversed when the plant is watered. This occurs when the turgor pressure in the non-lignified plant cell falls to zero as a result of diminished water in the cells. The rate of loss of water in the plant is greater than absorption of the water in the plant. So the process of wilting modified the leaf angle distribution to become much more erectophile conditions as opposed to planophile, meaning that it is 90 degrees away from the sun. So wilting diminished the plant's ability to transpire and grow. Permanent wilting is dangerous because it causes death. Aquaporins. Water molecules are small enough to move across the lipid bilayer of the plasma membrane, but they also move through specific channels of the passive diffusion of water across the membrane, which is called aquaporins. Aquaporins do not affect the direction of water flow, but rather the rate of diffusion. There are three major compartments of vacuolated plant cells. First of all, you have the cell wall, then you have the cytosol, and the vacuole. For the vacuole, it is a large organelle that can occupy as much as 90% of the protoplast volume. So they have a membrane called the vacuolar membrane, which regulates transport between cytosol and the vacuole. Between cell to cell, there is a continuity, meaning that actually, despite having walls between each other, each cells are actually connected. So you have the cytoplasmic continuum called symplast, and also you have the cell wall continuum called apoplast. So the short distance transport of water in tissues and organs, there are three routes. First of all is out of one cell across cell wall into another cell. They call it the transmembrane route. Second will be via the symplast or symplastic route. If you can see there, symplast route will be from cytoplasm to another cytoplasm through this wall, to the hole in the wall called plasmodesmata. And then the last one will be along the apoplast or apoplastic route, meaning that they are traveling through the wall. The movement of fluid in vessels is called bulk flow. In bulk flow, movement of fluid in xylem and phloem is driven by the pressure differences at the opposite ends of the xylem vessels and sieve tubes. Transpiration, which is evaporation of water from the leaves, reduces pressure in the leaf xylem. This creates tension that pulls materials out the xylem. In phloem, hydrostatic pressure is generated at one end of the sieve tube, which forces sap to the other end of the tube. So as you can see, there is two places. One is the sink and one is the source. So let's say that the leaves, they perform photosynthesis. There'll be a lot of sugar compared to the other part of the tree. There'll be less sugar because there's kind of like less photosynthesis. So you have the gradients. In the source you have a lot of sugar, in the sink you have less sugar. So the sugar will move from the area where it's concentrated to the area that is less concentrated or dilute. Roots absorb water and minerals from the soil. Water and mineral salts from the soil enter the plant through epidermis of roots and ultimately flow through the shoot system. So it goes from soil solution, root hair, epidermis, root cortex, and the root xylem. So in the root hairs, it is where much of the absorption of water happens, where the epidermis is permeable to water and where root hairs are located. Root hairs account for much of the surface area of the roots. lateral transport of minerals and water in roots. So, in one, you can see that uptake of soil solution by hydrophilic walls of root hairs provide access to apoplast. Water minerals can then soak into the cortex along the matrix of the walls. And then it goes to the plasma membrane. When the water crosses the plasma membrane through the symplast. As soil solution moves along the apoplast, some water minerals were transported into the protobulb cell of the epidermis and the cortex, and they're moving once via the symplast, meaning that they are moving from cytoplasm to cytoplasm. Within the transverse and radial walls of the endodermal cell is the Casparian strip, a belt of waxy material which blocks the passage of water and dissolved minerals. Only minerals in the symplast or entering the pathway by plasma membrane of the endodermal cell can detour around Casparian strip and pass into the vascular cylinder. Endodermal cell and also parenchyma cells within the vascular cylinder discharge water and minerals into the walls, which is called apoplast. The xylem vessel transport the water and minerals upward into the shoot xylem. Mycorrhizae. Mycorrhizae is a symbiosis between fungus and the roots. So both of them are actually two different beings working together. So the white mycelium around the fungus would provide large surface area for absorption of water and minerals from the soil. The endodermis. The endodermis will be the innermost layer of cells within the root cortex. It surrounds the vascular cylinder and functions as the last checkpoint for selective passage of minerals from the cortex into vascular tissue. Water can cross the cortex via symplast or apoplast. The waxy Casparian strip of the endodermal wall blocks apoplastic transfer but not symplastic of water and minerals from the cortex to the vascular cylinder. Plants lose an enormous amount of water through transpiration, and transparent water must be replaced by water transported up from the roots. Xylem sap rises to heights of more than 100 meters in tallest plants. So as you can see here, the roots will take in more water. Water is pulled out to the xylem. Water is replaced for inner cells. And lastly, water leaves to stomata and evaporates. So the movement of water is from the ground all the way to the air to the top of the tree or the plant. Pushing xylem sap, root pressure. At night, when transpiration is very low, root cells continue pumping mineral ions into the xylem of vascular cylinder, which lowers water potential. Water flows in from the root cortex, generating pressure that forces fluid up to the xylem. So this is called the root pressure. Root pressure sometimes results in guttation, which is the exudation of water droplets on tips of grass blades or the leaf margins of some small herbaceous dicots in the morning. More water enters the leaves than it is transpired, and the excess is forced out of the leaf. So, the mechanism that is used for transporting water is called the transpiration, cohesion, tension mechanism. So the first part of this mechanism will be transpirational-pull. So water vapor in the aspects of the leaf diffuses down to the water potential gradients and exits the leaf via stomata. Transpiration produces negative pressure, tension, in the leaf, which exerts a pulling force on the water in the xylem, pulling water into the leaf. The transpiration-pull on xylem sap is transmitted all the way from the leaf to the root tips and even to the soil solution through cohesion and adhesion, because water molecules, they tend to stick to each other through hydrogen bonds and also stick to a surface, so it actually adds to the strength. Stomata help regulate the rate of transpiration. Leaves generally have broad surface areas and high surface to volume ratios. These characteristics increase photosynthesis and increase water loss through stomata. The effects of transpiration on wilting and leaf temperature. Plants lose large amount of water by transpiration. If the lost water is not replaced by absorption through the roots, the plant will lose water and wilt. Respiration also results in evaporative cooling, which can lower the temperature of the leaf and prevent the denaturation of various enzymes involved in photosynthesis and other metabolic processes. About 90% of the water a plant loses escapes through stomata. Each stomata is flanked by gut cells which control the diameter of stomata by changing the shape. Changes in turgor pressure that open and close stomata result primarily from the reversible uptake and loss of potassium cells in the gut cells. So, transport of potassium ions across the plasma membrane and vacuolar membrane causes the coat changes of the gut cells. So, cells turgid, stomata open. Cells flaccid, stomata will close. Xerophyte adaptations that reduce transpiration. So xerophytes, they are plants that are adapted to arid climates, meaning that very extreme conditions such as a desert. So they have various leaf modifications that reduce the rate of transpiration. The stomata of xerophytes will be concentrated on the lower leaf surface and often located in depressions that shelter the pores from the dry wind. Organic nutrients are transported through the phloem. Translocation is the transport of organic nutrients in the plants. So phloem sap is an aqueous solution that is mostly sucrose, travel from sugar source to a sugar sink. A sugar source is a plant organ that is a net producer of sugar, such as mature leaves. A sugar sink is an organ that is a net consumer or storer of sugar, such as a tuber or bulb. Seasonal changes in translocation. A storage organ such as a tuber or bulb may be a sugar sink in summer, as it stockpiles carbohydrates. After breaking dormancy in the spring, this storage organ might also become a storage. After breaking dormancy in the spring, the storage organ may become a source as its starch is broken down to sugar. So it is carried away in the phloem to the growing buds of the shoot system. For example, the sugar maple. In the very early spring, it hydrolyzes the starches stored in its roots into sugar. This causes water to pass by osmosis through the endodermis into the xylem ducts. The continuous inflow forces the sap up the ducts. Phloem loading. Phloem loading results in high solute concentration at the source end of the plant. This creates hypnotic conditions in the phloem, causing water to flow into the phloem. Hydrostatic pressure builds in the sieve tube, but it is greatest in the source. At the sink, osmosis occurs with the unloading of sugar. Water flows out of the phloem. Build-up of pressure at the source and reduction of pressure in the sink causes water to flow from source to the sink, carrying the sugar along with it. Water is recycled via transport in the xylem. The explanation, it is very simplified. Scientists are just now discovering the details. This explanation is very simplified. Scientists are just now discovering the subtle details of the flow movement in plants. In many plants, phloem loading requires active transport. Means that you need proton, you need energy. So proton pumping and co-transport sucrose and H+ enables the cells to accumulate sucrose. This loading and unloading results in mass flow of substances in the phloem. H+ sucrose pump. First, sucrose is loaded into the phloem via a chemiosmotic ATPase mechanism coupled by H+ sucrose steam port. Other active and transport mechanisms, which is H+ and sucrose pump. H+ is actively pumped out by the hydrolyzing ATP. H+ is accumulated outside the membrane, generating a concentration and electrochemical gradient. H+ cannot cross the membrane, but there is a carrier protein. H+ binds to carrier protein, but sucrose must also bind. When both are bound, the configuration changes and the protein opens to the membrane interior. Downstream, sucrose must be unloaded, again utilizing a H+ sucrose pump. What is the proof to support this? The rate of flow in the phloem is about 10,000 times faster than it would be if it was only due to diffusion. pH of the phloem sap is around 8, so it is alkaline due to the loss of hydrogen ions. There is an electrical potential difference across the cell surface, which is negative inside, due to presumably the loss of positively charged ions. So recap, in this chapter we learn about 3 types of transport in plants: membrane selective permeability, bulk flow of phloem and xylem, and also xylem pressure and phloem translocation. The end. I'll meet you in the next chapter.